your lore master himself, Mr. Rawlins. I've got DNA behind me. Today we're going to be taking a look at 16.3. That's right, we're looking at DNA replication. Let's get to it. I'm going to share my screen. We're going to take a look at the content focus report real quick, because it's important to strategize when you're preparing for a test. Understand what it is that you're being asked and understand what it is that you need to know. So 16.3, we're describing the basic process of DNA replication, how that relates to transmitting DNA from one organism to the next, and how we conserve that genetic information from time to time. We're also going to be taking a drop down here to look at benchmark clarifications. We need to describe the process of DNA replication or its role in transmission and conservation. Items requiring content and limits. This is important too. Items requiring the analysis of base pairs for gene mutations are limited to changes in a single gene. And I think that's all we need to know in regards to DNA replication. Now, on the past of the EOCTs, you'll notice that in 2019, there was one question on DNA replication. 2018, same thing. But some of the topics that we'll get into with DNA replication affect the others as well. It'll affect how well you understand things like transcription as we turn from DNA into RNA. It'll affect how well you understand what a mutation is along the way as we talk about DNA replicating itself. It'll even extend into other standards like mitosis, meiosis, when we're talking about interphase and replication of DNA there. So this kind of gets spread all over the place like all the standards in biology do. They all come back to evolution, moving on your genes, and making adaptations to your environment. Let's swap a Ruski over into my whiteboard. I've got prepared for you the 4 nucleotides: adenine, thymine, cytosine, guanine. If you recall from standard 18.1 on macromolecules, nucleotides are the monomers that are put together to make our nucleic acids like DNA, RNA, and others. So here's our batch of nucleotides that we can piece together to make DNA. At the top, we've got 4 in a row that have been connected: ATCG, that's adenine, thymine, cytosine, guanine. I've grouped them like this in a couple of ways, a couple of reasons. For one, the A's and T's are always going to go together, adenine and thymine, and our C's and G's are always going to pair up together. They fit well together, attracted to each other, and bond well whenever we're making a double-stranded DNA molecule. So in our first molecule, you notice it's unfinished. We only have one side full of our monomers, the nucleotides. We need to finish up the other side. So the other side, based on our base pairing rule of cytosine with guanine, adenine with thymine, DNA carries 2 strands with it, and they need to be paired up. So our A's and T's go together, adenine bonds to thymine. Our thymine bonds with adenine. Our cytosine is going to go with guanine, C's and G's, and guanine with cytosine. Now this is, say we've got a cell just blubbering around, chilling in interphase. This is the DNA that's inside of it. DNA is the instructions that tells the cell, hey, Mr. Cell, here's some instructions on how you can build proteins too. It's important. And we need to pass that DNA instruction on to the next generation. So how do we do that? In interphase, part of the cell cycle piece, this DNA is getting replicated. Replicated is the official word. It means it's getting copied. And we would hope that we have perfect copies. To make that happen, there's a set of proteins. Proteins do all the cool stuff in your cells for the most part. In this case, we actually have an enzyme that's speeding up reaction rates. See, I'm pulling all kinds of standards, going back to 1811. And so there's an enzyme that's basically scissoring in half our strands and going to cut them into 2. So our left strand's going to go over here, our right strand's going to go over here. And there's even more enzymes that come along to take our original strands, ATCG on the left, and T, A, G, G, C on the right. And there's other enzymes that are going to come along, little proteins, that are going to reassemble the matching set for both of these strands. So coming along, first one to go in, matching up with our A is a T. Next one comes along, gets sealed together. To match it with the T, the enzyme's going to pull an A. Next one coming along, our enzyme's going to pull another nucleotide to make this polymer, from monomer to polymer. C and G's match up. And then finally, once again, C's and G's match up. TAGC. What you'll notice is that ideally, if we're doing perfect replication, the new strand that we just built to match the left side should be the same as the other strand that matches the left side from up top. That's what we're trying to do. We're trying to make perfect copies of the original strand. So matching A's with T's, adenines with thymines, thymines with adenines, cytosines with guanines, and cytosines with guanines. Boom. We should have now 2 sets of DNA. And so our original cell that was wanting to pass on its genetics can now pinch in 2 through the phases of mitosis and create 2 identical cells. Aw, how cute. So that's DNA replication. Moving into some other standards from 16.3. There's also the issue where we can have some of these nucleotides not pop into the right spot. So maybe when it's getting replicated, another letter jumps in. And this strand all of a sudden has another A and another T. That's an insertion mutation. There's all kinds of different ones. There's deletions. Maybe we lost original letters. Maybe more than one letter comes in, but if you'll recall, that's too far for high school biology. Just one letter at a time. And so those mutations, as they add more nucleotides, as they add more letters, are going to change what that DNA reads out. It's going to change the gene that it's coding for. What does that mean? Is that good or bad? It just depends how it works out. In the case of the polar bears that we saw, when they had a broken gene that no longer let them grow the brown fur pigment, it actually helped them out for a while because they were better matched to snowy, icy environments. They could camouflage better. But nowadays, in a world of global warming, polar bears are actually struggling really bad. We'll probably see the extinction of polar bears in our lifetimes because polar bears just don't have the ice to blend in anymore. So mutations are neither good or bad. It just depends how they match up with your surroundings and if they help you survive or not. All right, so that is DNA replication in a nutshell. See you next time on Short Science with Rawlins.